Bismillahirrahmanirrahim. Assalamu alaikum dear cadets. Welcome to the new lecture. Today our topic is lysosome, another cytoplasmic organelle. Lysosome is 0.2 to 0.5 micrometer in diameter, meaning the term lysosome is derived from lyso means lysis or splitting or breaking, while soma means body. Why it is called lysis or splitting or breaking? When we will study its function, so you will understand that why it is why the lyso refers to lysis. Discovery: lysosome was first discovered by a Belgian scientist Didieu in 1949. Definition: how can we define lysosomes? Lysosomes are small sacs or cytoplasmic vacuoles bounded by a single membrane. Lysosomes are small sacs or cytoplasmic vacuoles bounded by a single membrane. The membrane is impermeable to outward and inward flow of substances. Occurrence: lysosomes are present in the cytoplasm of eukaryotic cells, particularly in animal cells that exhibit or show endocytosis. That is, lysosomes are abundant in the animal cells which are involved in phagocytic activity, For example, macrophages and neutrophils, which are the two types of white blood cells that are involved in phagocytosis. Formation: how lysosome is formed. Lysosome contains numerous hydrolytic and acid phosphatase enzymes. These enzymes are synthesized in rough endoplasmic reticulum, then transported to Golgi bodies or Golgi apparatus for further chemical process. Here the enzymes are packed in membranous vesicles. Lysosomes contains numerous hydrolytic, which performs hydrolysis, breaking of macromolecules. Lysosome contain numerous hydrolytic and acid phosphatase enzymes. These enzymes are synthesized in rough endoplasmic reticulum, then transported to Golgi apparatus for further chemical process. Here the enzymes are packed in membranous vesicles. The processed enzymes are budded off as Golgi vesicle, and these vesicles are called primary lysosomes. Once a lysosome has fused with a vesicle containing the material to be digested, the structure is called secondary lysosome. We will now explain this formation with the help of a diagram. Now if you look into this diagram, this is a simple diagram showing rough endoplasmic reticulum, Golgi apparatus. These are the cisternae of Golgi apparatus. These are transport vesicles coming from rough endoplasmic reticulum to Golgi apparatus. And this is cis phase of the Golgi apparatus and trans phase, and this is plasma membrane or cell membrane. So as you studied earlier, that lysosome contain many enzymes. These enzymes are synthesized in rough endoplasmic reticulum and then transported, look into the arrow, transported to the Golgi apparatus for further modification or chemical process. After modification, here these enzymes are packed into membranous vesicles. The processed enzymes are budded off as Golgi vesicles, and these vesicles are called primary lysosomes. The processed enzymes are budded off as Golgi vesicle. This is Golgi vesicle, and now this vesicle is called primary lysosome. Again, lysosome contain, this is primary lysosome. The lysosome contain many digestive enzymes. These enzymes are synthesized in rough endoplasmic reticulum and are transported to the Golgi apparatus for further chemical process. Here the enzymes are packed in small membranous vesicles and the processed enzymes buried up as Golgi vesicle, and now these Golgi vesicles containing digestive enzymes are called as primary lysosome. This is how lysosome is formed. Now chemical composition of lysosome. Chemical composition or enzymes of lysosome. Lysosome contain a very large number of food digesting enzymes called hydrolysis. Food digesting enzymes called hydrolysis. It contains about 40 different digestive enzymes such as proteases, nucleases, RNases which break RNA, DNases which break DNA, proteases break down or digest proteins, lipases lipids, glycosidases carbohydrates, phosphatases phosphate group, phosphate bond. The content of lysosome is acidic. That is why acid phosphatase enzymes means, because the content of lysosome is acidic, that it has low pH, that is 1.5 pH. The content of lysosome is acidic, that is it has low pH of 1.5. These enzymes break down every major macromolecule of the cell such as proteins, nucleic acids, lipids or carbohydrates, phosphate, etc. Now functions of lysosome. The first function is phagocytosis, or when the material taken in by endocytosis is large. You know that endocytosis is of two types: phagocytosis, cell eating process, and pinocytosis, cell drinking process. So when the material is solid, it is called phagocytosis or cell eating process, and when it is liquid, then it is called pinocytosis or cell drinking process. So when the material taken in by endocytosis is large, such as food particle or another cell, the process is called phagocytosis. When the phagocytic vacuole fused with a lysosome, digestion occurs. This is how mammalian white blood cells engulf and destroy bacteria and other cells. So this is the first function of lysosome, phagocytosis, or function in immune system or cellular digestive system, because this is how mammals white blood cell engulf and destroy bacteria and other cells. We will also explain or study these functions with the help of a diagram later on. Another function of lysosome is that autophagy. Auto means self, phagy means eating. So autophagy means self-eating. What is autophagy? The process by which unwanted structures in the cell are engulfed and digested within the lysosomes is called autophagy. The process by which unwanted structures in the cells are engulfed and digested within the lysosomes is called autophagy. During autophagy, old, worn-out parts are also digested. In this way, materials of the cell may be recycled and cell may be renewed. So this function is also known as renewal of cells or recycling of materials. That is, lysosome act as scavengers and ingesting and digesting worn-out parts of the cell. Because of this activity, the lysosomes are also called as garbage disposal units of the cell. For example, a human liver cell recycles half of its macromolecules each week. Or for example, when the mitochondria become old, functionless, lysosome fuse with it, break it and recycle nutrients. So this is called autophagy, or self-eating or renewal of cells or recycling of materials. Another function of lysosome is autolysis. Auto means self, lysis means digestion, self-digestion. What is autolysis? Autolysis is the self-digestion of a cell by releasing the contents of the lysosome within the cell. The self-digestion of a cell by releasing the contents of lysosome within the cell. In such circumstances, lysosomes has been named as suicidal bags, and the name of lysosome comes from here. Lyso means splitting and soma means body. As at lysosomes act as suicidal bags. Autolysis is a normal event of developmental process. For example, removal of tadpole, larva. What is tadpole? Larva of frog. What is larva? Immature form of an animal is called larva. Removal of tadpole tail during metamorphosis. Metamorphosis means the change of shape during an animal's life. For example, a frog develops, hatches from an egg, then tadpole, then froglet, frog. So the change of shape during an animal's life is known as metamorphosis. So the removal of tadpole tail during metamorphosis is due to autolysis, which is performed by lysosome. Similarly, the removal of whip from human fingers during apoptosis, during development, is also an example of autolysis. Another function is exocytosis. Sometimes the enzymes of lysosomes are released. The enzymes of lysosomes are released from the cell by exocytosis. For example, during the replacement of cartilage by bone during development, lysosomal enzymes are released by exocytosis. Sperm also contain a special lysosome called acrosome. This releases enzymes for digesting path to the ovum or egg.